We started our webinar one with asking a question, which is we wanted really to insist on the importance of being very clear about what you want to know about your material and the material that you send to us, because simply the question to be answered will determine the analytical technique or techniques to be used. In this case, we'll be focused, as Gareth said, on the electron microscopy, infrared, and XRD, X-ray diffraction. So we started with this slide last time, and we finished up with this one, which is an illustration of the strengths of each technique that we want to present. So maybe just to summarize, basically you can see on this triangle that every sample that we receive will sit in this triangle with three poles: the solids, the water, and the oil, and anything in between will be mixed up. And the strength of each technique is being illustrated here, highlighted by a intensity of color. So for SEM, for instance, it is mostly solids that can be analyzed, but we've got a tinge here of light blue as well, which means that we can actually look at semi-solids and liquids when we use particularly the cryogenic attachment to the SEM. For XRD, it's essentially solids that that can be analyzed, and as far as FTIR is concerned, well, it's a very versatile technique, and we can get information about all types of materials, solids or liquids or semi-liquids. And of course, what is most important is the interaction between the three techniques and how the expertise is delivered by being interactive. So first of all, the produced water sample, and we start with the question. What do you want to know about that produced water? Are you interested in characterize the solids that they contain? And if so, how much material have you got available? In this case, we have filtered some solids, and typically it's on a membrane that is 1 to 2 inches in diameter. So you can imagine that this is quite a small amount of solids. So the first technique that we will be proposing to you is the scanning electron microscopy coupled with the energy dispersive spectroscopy. And we have mounted the sample onto an SEM stub, and we're looking at it first of all at very low magnification. And we're using both the morphology of the particles and the EDS analysis, the elemental composition, to determine, the type of material that we've got here. And we can say that here, for instance, we've got a larger particle that we have potentially identified as a plagioclase feldspar called albite, and another particle here is probably a quartz. But we are most interested in this case about these tiny little particles here, which by EDS analysis, the elemental composition gives us barium, strontium, sulfur, oxygen, and we're making abstraction of the fluorine and carbon in this case, because it really basically comes from the membrane that has been mounted at the same time with the sample. So this elemental composition guides us into a potential barium sulfide mixed up with strontium sulfate, or possibly a mixture of barium strontium sulfate. So when we look at higher magnification with the SEM, we can see that the particles are actually very well crystallized. They're tiny. This is 1 micron scale bar. They're very small, but they're very, very well crystallized. And the combination of this morphology and the EDS, the elemental signature, is a very strong indication that these particles that are tiny here are a newly crystallized precipitate of barium strontium sulfate. So this is the first indication using the SEM and EDS analysis. Now, do you need, still on the solids, do you need more information? For instance, would you like to know whether there are also some organics present, in which case we will send the sample to FTIR? Or would you like an absolute definitive identification of those solids? And XRPD will help you with that. So first of all, we'll send the sample to FTIR, and I'll pass over to Jean.
Okay, so when we get the sample like this on a filter paper, often if it's very small amounts of material, we have to run it directly on the filter paper. And mostly we find that if the material is not thick enough, we will get bands from the material on the filter, but also from the filter paper. So to get around that, what we would do would be to record a spectrum of the filter paper at a cleaner area, along with the one in an area covered in the material, and then we do an electronic subtraction and we get what we call a different spectrum. So this is an example here from the sample we're looking at. And here we can see that there are bands related to organic material. We see CH2 bands, which tell us that there's long chain hydrocarbons, but we also see bands related to amide, looks like protein, which are indicative of often of a biological material. And the hydrocarbons could relate to the biological material because some fungal species have a lot of these long chain hydrocarbons, or it might be two different parts. But we're able to look at what's on the filter and assess what organic material is there.
Thank you, Jean. And then we send the sample to XRD. Steve, if you can take it on there. Steve, you're muted, I think.
Thank you. Yeah, well, by XRD, again, we see mainly diffraction from the filter itself, but we can see some very small peaks on there. And by running a filter blank as well, which is the red trace there, again, like the IR, we can do a subtraction. Next slide, please. Doing that, these are very, very tiny peaks, not very many counts. But it's very clear when we compare it to samples in the powder diffraction file that we have peaks which are intermediate between the position for a pure barite or a pure celestine, which is the strontium sulfate. And so it very definitely looks like we have a solid solution. Next slide, please. And we can go a little bit further and actually estimate the composition of the solid from the precise positions of the peaks in that pattern.
Okay, thank you, Steve. So you can see that we had very little material, but using the three techniques, we were able to get quite a lot of information at different levels. So next, still with the produced water and still with the solids on this produced water, but this time we have solids that have actually dropped out in a flask overnight, and we have a little bit more of material. We've got larger amounts, therefore our first protocol tends to be with XRPD because they do need in most of the time a little bit more material. So we thought that maybe that would be the best first technique to use. Steve, that's back to you.
Yeah, thank you. Yeah, well, I think for this particular diffraction pattern, probably about 0.5 gram of the dried solid was placed into a cavity holder in contrast to the milligrams that we were looking at on the filter. But in this particular case, there are no distinct sharp peaks at all in the pattern. All we can see are two kind of broad scattering maxima, one of them centered there at a d-spacing that's in angstroms of about 2.6. So clearly by XRD, we have some kind of amorphous, X-ray amorphous material here, which basically means there's no long-range crystalline order in the material. A glass, for example, a window glass would give a diffraction pattern that's not dissimilar to this. So not a great deal of information at the moment from XRD other than that it's X-ray amorphous.
Okay, thank you, Steve. And with that, we just thought that the best was to send it to FTIR. So Jean will just be able to take it from there.
So the FTIR does have the advantage that we can do organics and inorganics, but we can particularly do inorganics, which are either crystalline or amorphous, because we get bands which relate to the structure that we can see, even if they are amorphous. So in this particular case, what we see is that it's the spectrum is dominated by hydroxyl bands, so there's sort of strong kind of hydration present. We can identify that there's some organic matter there, but the other inorganic bands which appear to be there would be consistent with the silicate, an amorphous silicate, and probably iron rich. Now, we can't be completely sure because bands around about the area where you get the SiO stretching for the silicate can also be carbon oxygen or phosphorus oxygen. So sometimes if we're not entirely sure, we need to get some backup from elemental analysis, and this is where we pass it back to SEM EDS to try and identify to try and confirm whether what we suspect is backed up by the elemental analysis and see whether any other cations are present.
Okay, so back to SEM, and we took an image of the material. It's really nondescript, it's amorphous, so it's very difficult to get anything out of that. But the EDS analysis is very clear. We do have iron and we do have silicon. So basically, the combined analysis of the XRPD, we determined first that it was amorphous. The FTIR, we just had a fairly good idea of what was in there. And the confirmation by SEM EDS just give us this identification of an amorphous iron silicate for this sample. So next case study is on a core sample. So it's core material. This is a plug taken from a core sample. It's a sandstone. And yeah, first question again, what do you want to know? Often the very first question will be on what is the mineralogical composition, and there are several ways we can go by there. We can ask for a qualitative, simply mineralogical composition. We can ask for a quantitative, or we can maybe ask whether there is specific clays that are present. It's very important, for instance, for swelling clays, if they are there or not. In any case, any of these questions can be answered by XRPD. Steve, you?
Thanks, Jean. Yeah, so the first thing we would do is usually run a whole rock or a bulk X-ray diffraction pattern of the material. That goes through the first phase of qualitative identification of the minerals present, and then moves on to the quantitative analysis. And so here's an example for this particular sandstone here, where we identify a range of minerals: quartz, plagioclase, potassium feldspars, some carbonates, pyrite, a little bit of barite, perhaps for some drilling mud. This is an oil industry sample, and some clay minerals. And this quantitative analysis is usually done these days by what's called a full pattern fitting method, which can give pretty accurate analysis of all and any of the minerals present in that sandstone. Next slide, please. Very often in the petroleum industry, clay minerals are a particular interest. And to get more detail on the clay minerals, we would typically separate them as a clay size fraction, less than 2 micron, for example, and then record a series of X-ray diffraction patterns following different treatments. These treatments are shown here in the different colors, and it's the sum of that information and how the different peaks respond to the different treatments. The heating at 550, for example, where you can see this big peak around about 7 angstroms, has disappeared. That sum of information tells us what clay minerals are in the sample, and also there are methods for quantifying these what are called one-dimensional diffraction patterns of these clay data by modeling the diffraction patterns and looking at things like relative peak areas. So a lot of information on the mineralogy from X-ray pattern diffraction.
Okay, thanks, Steve. Right, so we've got the mineralogical composition, but there might be other things that you might be interested in. You may want to know what the minerals themselves look like. What is their morphology? For clays, for example, is the clay fibrous or is it platy? What is the distribution of the various minerals. Are they found in a small pocket, or are they widely spread throughout the sample? This is particularly important for the swelling clays, for example, or for iron-bearing minerals like iron chloride, which may affect the conductivity readings of the formation. You might also be interested in the diagenesis of the sediment, the order of precipitation, how the minerals interact, how the minerals have been replaced, or more importantly, you may be interested in the porosity of the sandstone. What are the pore types, their geometry? Are they micropores, micropores? What the size of the pore throats? And the distribution, is the porosity evenly distributed, or does it follow some preferential channels? And the techniques for all this is very visualizing, and it is helped by scanning electron microscopy. With the help of energy dispersive, the EDS, to know where the minerals are identified. And if we know the mineralogy of the sediment by using their elemental composition, we can find out where those minerals are. So some examples here of importance of the variations of the mineral morphology. We do have some clay minerals here. Sorry, I think I've lost the pointer here. We do have some clay minerals like some kaolinite here that is filling a space, a pore space between two quartz grains and some other quartz crystals. But if we look here, there is also another platy clay, and that platy clay is there, so it's a completely different distribution. This one is a grain coating clay, so very important. And this is chlorite, a platy clay, and this is kaolinite, another platy clay. You can see that within the kaolinite we do have some microporosity as well. Another type of clay is with a fibrous morphology. This time is elite, and you can see that this is a sand grain. This is another sand grain, and you can see that the space between the pores and the pore space between the two is bridged by these fibers. So very important to know how this. Sorry, I just can't see. There you go. The pores, the porosity itself, whether we have macropores or micropores. In this example, we have two different sandstones taken at the same magnification. So if you can look at the size of a sand grain here, this is more or less the same size. So we've got very similar sediment in terms of grain size. But the difference is the pore space in between all the grains. Here we have very large open pores, possibly up to 100 micron in size, where here the pores have been filled with this granular material, which is it's actually kaolinite clay. So the difference between the macroporosity and the microporosity is that here we're going to have a very, very good permeability of the sandstone, whereas here the flow is going to be a lot slower, much lower permeability. Another difference in terms of porosity is primary pores versus secondary pores that come from the dissolution, in this case, of some feldspar grains. So this is all things that you might be interested in and that we can provide. So by combining the mineralogical characterization from the two different techniques, we can see very clearly that XRPD will identify and quantify the minerals, whether the SEM will actually visualize everything and help understanding how they interact. Our next sample, our next case study is on a sludge sample, something that is very sticky and usually very smelly. Again, the question is, what do you want to know? Is that a quick evaluation of the composition, or are you interested more in the presence of or absence of a particular compound? In any case, the first protocol here is going to be a FTIR. So I'll pass you over to Jane.
Now, when we receive this type of sludge sample, and they are quite common because sludges can form in so many different places and they almost invariably cause a problem blocking things or causing other issues. And we always take a sludge and we'll run it, the FTIR, as received. However, if, as many sludges do, there are hydrocarbons present, then you can find that this dominates the spectrum. And so it means that we can't just rely on the spectrum in order to work out what's there. We've got to do some preparation, and often it's a solvent extraction, which we generally do with dichloromethane, DCM, because it's both a good solvent for many organics, and also it rapidly evaporates so that we can record our FTIR. So in this particular case, we see it's dominated by hydrocarbons. There's some evidence that there's paraffin wax there, but also other hydrocarbons. So we would therefore go and do an extraction. So as you can see, we've got our original spectrum at the top, we've done a solvent extraction, and we've got the insoluble solids out, and we've done the spectrum of them. And as you can see, they're very different, completely different from what we were originally seeing. So the preparation is very important. Looking at this, we can see that the main bands present are a carboxylate. There are evidence that maybe other things such as sulfate present, but the main thing present is this carboxylate. And looking at the frequencies of it and comparing it with reference spectra, it suggests that it's a polyacrylate salt. And we can be fairly confident it's this type of material, but what we don't necessarily know is what the cation is that's present in that salt, and that might be important for the customer. So in that particular case, we take it as far as we could, and we'll pass it back to EDS for identification of the cation. Now, the EDS trace that we've got here shows us the high C and O, which come from the polymer, and they show the dominant cation is calcium, although there's also magnesium and sodium there. So we can therefore put that together with the information we've had from the FTIR and say that as well as the hydrocarbons in the sludge, the dominant compound is a polyacrylate salt, probably predominantly calcium polyacrylate, though potentially, as I say, magnesium and sodium in there as well. And it may be, we can sometimes have an idea from the customer as to what's in the system. So if there's a polyacrylate scale inhibitor present, then it could be that there's a reaction with a calcium source, which is causing this intractable sludge to form. And another case study, which uses FTIR, is a different kind of thing where we're looking at compatibility of injected fluids and umbilical liners. Where you're going to have a fluid sitting in a pipe with this type of liner, it's very important that the client needs to know, is there going to be any interaction between the fluid and the liner, because this could potentially cause fluid contamination. And also, if something's coming out of the liner, then potentially could also be causing degradation. So in order to study that, we take a look at the FTIR of the fluids. Now, what we start with is the control fluid. So that's the fluid which has completely clean, it's not been in contact with the liner at all. And then generally we compare it with an aged fluid. That's a fluid which has been placed in contact with the liner for some time, often at elevated temperatures, because that speeds up any kind of reactions that might be going on. So when we look at this comparison between the two, we can pick up the additional bands which are appearing in the aged fluid, the ones with the P's beside them. And we know that these bands are identifiable as belonging to a plasticizer. So we've identified that there is a plasticizer in the aged fluid, and that is obviously both contaminating the fluid, but probably more importantly, indicating that there will be degradation of the liner. But sometimes for a customer, they need to know exactly how much plasticizer is leaking, because maybe tiny amounts are not going to cause degradation, but a larger amount might fall outside the range which is acceptable. So in that case, we're not set up to do quantification by FTIR, so we pass it on to colleagues in the wider analytical services that we offer, who do gas chromatography. So they're able to take they will do an extraction in the same way. They're able to do the quantification and compare it by comparing it to an internal standard. So then, as I say, we have a full picture to put back to we can put back to the customer, and they will be able to know whether the plasticizer, which is being leached, is being leached to such an extent that there's going to be a problem.
Thank you, Jean. And then the last part is by doing a scanning electron microscopy on the liners themselves. So comparing with, again, a control liner and the liner that had been used for the testing. And you can see here that from a smooth surface, we have some kind of paving, crazy paving texture here, which at higher magnification shows that it's definitely cracked. So the liner itself has definitely been damaged and degraded, and you know, giving more time, it probably would crack completely and completely fail. That is it for our case studies. I'll pass you over to Gareth to wrap up.
Okay, thanks Evelyn, and thanks very much Jean and Steve for the contributions as well. That was extremely informative. I'm just going to say a few things to finish off. While I'm saying that, if you have any more questions that you'd like to ask, now's the time to pop a few down in the questions and answers. We have a few, so I'll come to those in a minute. Just to emphasize with the slide on the screen at the moment that there are other staff involved in these three techniques as well. And if you need to get in touch with SEM, then you can see the team there. FTIR and XRPD, everybody is named on the screen. Jean indicated we do other techniques as well. We do have a wide variety of other analytical techniques. For inorganic analysis, we have ICP, MS, and OES. And we also have a fusion technique to put into solution if we want to quantitatively analyze something by those techniques that isn't readily something you can dissolve in standard mineral acids. Then we can use a fusion techniques to put stuff like silicates into solution and quantitatively analyze them. We have ion chromatography, discrete analysis, TOC, TN, quantitative organic analysis, GCMS for volatiles and semi-volatiles, GCFID. For heavier organics, we've got things like LC-MS-MS and HPLC. And we also have an isotope section, continuous flow isotope ratio, mass spectroscopy, carbon-13, carbon-12 ratios, and N15. And quite uniquely, well, probably not uniquely, but rare, we do have a thermal ionization mass spectrometer as well for doing strontium, neodymium, samarium ratios and strontium isotopes for stratigraphy interpretations, plus many bench techniques, wet chemistry and physical techniques as well. Okay, so thank you very much for coming along to this webinar. I'll just run through some of the questions that we have and put back to the panelists. First question is to Steve on XRPD. How do you make a quantitative analysis by XRD, Steve?
Okay, yeah, there's various ways of doing it. The key thing is the sample preparation. You have to get that right. You have to ensure the sample is milled to a very fine particle size, microns, and you have to try to eliminate preferred orientation. The way we do it at the moment is by a full pattern fitting method. We have reference patterns for all the minerals that we might encounter in a sample, and we fit those by a least squares procedure. If you're dealing with more common compounds as well, the things that don't include clays, there's a method called the Rietveld method, which is widely applied to do quantitative analysis, and that's pretty good for most things, but can struggle quite a bit when you get clays involved. So basically, these days it's a fairly automated procedure that's using all the information in the diffraction pattern, but it's got a very, very long history actually. Quantification by X-ray diffraction goes back, I think, even to the 1930s, when people were first started using it for quantification of cements and things like that.
Okay, thank you very much,
Hopefully
Steve. Yeah. Another question that it goes to everybody, so perhaps I'll start with Evelyn. So the question to everybody is, how much sample do you need for each technique? So Evelyn, can you say that for SEM, please?
Yeah, well, SEM is basically, if you can see it, you can do it. Sometimes you can't even see it, but you can still do it. We really need very, very little material. What you need to ask yourself, though, is whether the sample that we are going to be analyzing is representative of your material. So that's the main question for you to decide. We have received some 5 kilos of material, and it's really for us very difficult to decide what do we want to look, because we're looking at a micron scale. So ideally we want small samples, but we can look at a much larger sample if necessary.
Thanks, Evelyn. Jean.
Hi, Gareth. Yeah, we can also work with relatively small amounts. I mean, usually, again, not obviously quite in the scale of the SEM, but we usually reckon that if we can actually see it and pick it up and get it onto the sampling accessory, we can analyze it. And we would usually say we'd like 1 milligram of material, but again, as Evelyn says, very important to be representative. And often for FTIR, what we can do is to grind small sort of inhomogeneous samples might be ground up and run as one, or particles might be picked out. But generally, FTIR can be recorded actually on very small samples. And because we can run the technique that we use for sampling allows us to put things directly on, we can do things like take bits of a solid or a polymer or something and just place it on directly without further preparation. So yeah, small amounts and relatively flexible is what we can do.
Thanks, Jane. And XRD?
Yeah, for XRD, I mean, typically we need a larger sample than the other two techniques, particularly if we want to do quantitative analysis. So we're into the kind of gram quantities normally. However, we can also look at very small samples. We have some special low background holders that will allow us to just sprinkle a couple of milligrams of something on there. The caveats with that is that some of the analysis then are restricted probably just to sort of identification of compounds rather than quantitative analysis. You can attempt quantitative analysis, but it does become more tricky when the amounts are very small. So typically for XRD, you know, we would hope that you would have samples of a couple of grams or something like that, but it can work with milligram quantities if needed.
I'm not quite sure who wants to pick up on this, so I'll just ask this question directly, and then whoever wants to start can start. How do you decide what technique you start with? Who wants to pick up on that?
I'll start up, and maybe you can add up onto that. Well, basically, this is back to what's your question? What do you want to know? But there are two things that we need to consider, and is the size of the sample and of course what type of material it is. So that's what we saw earlier on. If we want to get some solids and we've got very, very little, it might be that SEM and EDS is the very first one to look at. It's not going to give you a full absolutely positive identification, but it might be that's just what you want to know. If you have a lot more material, then maybe XRD is the best first techniques if you want to know exactly what the composition of the minerals you have. If it's a very sticky material like our sludge, for instance, or semi-liquids or liquids, certainly FTIR will be the first technique we'll go for. And if they need more information, then they'll come to us for other type of data that we can provide. Don't know if that's enough, Jean, Steve?
Yeah, no,
Yeah.
I think that's largely, as I say, it starts with the question and the amount of material and goes from there.
So does somebody decide that, or do you get together and decide what the first approach is, or?
Well, hopefully we would have a conversation with the customer when they first decide that they want some analysis to be done. What I think is very important is real good communication. So if we can have that discussion with the customer, saying what are you after, and then we can decide. And sometimes we need to wait until we see the sample before we can decide exactly which first technique to use, or what other techniques. It might very well be that us, SEM, FTIR, XRD, we can't do anything, in which case we'll pass it to some other of our colleagues in the institute. But yeah, that conversation to start with, I think, is crucial with any customer to find out exactly what they are after and how we can help them, because basically we're here to help. We're not here to do an analysis that just for the sake of doing the analysis. We've had the case before where we were sent some produced water and or some liquid, and we were asked do the analysis on that, but we were not told, you know, what are you interested in? Are you actually interested in the liquid in there, or are you interested in the solids within this material? And it's only by discussing with the customer that we find out. Otherwise, it's a complete waste of time.
Okay, thanks, Evelyn. So yeah, so clear communication is key. A question for FTIR. How can you analyze both organic and inorganic compounds?
Thanks, Gareth. Well, FTIR is very versatile, as Evelyn said, because, as I say, it basically, for vast majority, we can analyze most things. What we can't do are salts such as sodium chloride, although we can do them if they're hydrated. And the reason is that we need to have bonds between the atoms in order to for the infrared to be absorbed and to get the spectrum. But that can be for some solids within something like a sulfate. A sulfate will have sulfurs connected to oxygen, same with carbonates or silicates. So even though they're inorganic, they still will give the bands. The bands will be slightly different. They're not generally as sharp as you see in the organics, but in both cases, it's bonds between the atoms that are vibrating, absorbing the radiation. And as I say, for most inorganics, you actually have some of those bonds that can absorb the radiation as well as in the organics. As I say, there are certain salts where they're just purely ionic, purely electrostatic between the atoms, but even many of them are hydrated, and we can see the hydration pattern and say something about what we think will be there. So that's the way that we're able to do both of those.
Okay, thanks everybody. That's I trust the questions are answered in full. They certainly seem to be to me. If there's any follow-up, please indicate now in the questions and answers. That's all the questions I have for the moment. Does anybody want to be unmuted? If so, please indicate in the questions and answers or the chat, and we can unmute you and you can ask a question directly.
Gareth, there's a question in the chat at the
There's
moment.
a question in the chat. I don't see it. I'm not seeing a question in the chat. Can you ask it directly?
So the question's from Stuart Craig, and he says, for FTIR, how do you handle combinations of different compounds?
Okay, well, the fact is that when you've got a complex combination of things, then you get a complex combination of bands, and it's obviously yeah, it makes it much more difficult when you're looking at it, much more complicated. And there's two ways you can go about simplifying it. One is the preparation that we talk about, which is doing different solvent extractions. You can use water for certain things. You can use the DCM, as we've talked about, and that separates it out into different fractions, which means we have fewer bands and we see more clearly bands relating to different materials within it. But then within that, you have to look for key bands. You look for a key band which might indicate there's a phosphorus oxygen or something like that. But then you have to look for the bands that pick out bands that would go with it to confirm that that might be there. So it's a very complicated kind of process whereby you're using some of your knowledge about the material, you're using the band, you're looking at particular bands, but you're also looking for other bands to back up what you're doing, and then you can get reference spectra and compare it. So it's really complex mixtures are difficult, but it's a combination between the sample prep and recording different fractions and actually picking out different bits and putting it all together. And then at the end, you make sure that it all makes sense. And sometimes, as I say, we don't know whether it's perhaps phosphorus, oxygen, sulfur, oxygen, and then we'll pass it to Evelyn, or the team in the SEM, to do the EDS and tell us which elements are present, and then we go back and have another look at it. So it can be quite a long process and can be quite complicated.
Thank you very much, Jane. Just to emphasize, for some reason I'm not seeing the chat. I can see the questions and answers. There's another question popped up in the questions and answers from Gus. In XRPD, how do you quantify your clay fraction and how do you combine that into your bulk rock quantification?
Okay, yeah. Well, we quantify the clays in the bulk material directly. And what we get from the clay fraction are extra details about what those clays actually are. That's particularly so, for example, for mixed layer clays. For example, we might be able to quantify the presence of a mixed layer illite smectite in the bulk material, but only be able to determine the proportions of illite and smectite in that mixed layer clay from the clay fraction. So although we do quantify the clay fraction as well, we treat them really as separate quantifications. I have seen labs that try to combine these things, but you're really on a hiding to nothing. It's extremely difficult to combine, you know, one like extract a clay size fraction, quantify it, and then kind of add it back into the whole rock. It usually ends up with you know, pretty inaccurate results. So the best way is to focus on the whole rock or the bulk material, get the quantitative values for that from that, and get the detail of what the clays are from the clay size fraction.
Okay, thank you very much, Steve. Does that answer everybody's questions? And last chance if anybody wants to ask one from the floor. Laura, for some reason I'm not seeing the chat even when I click on the chat. Is there anything
There's nothing else there, Gareth.
Okay. So thank you everybody for joining today, and thank you again to the presenters for those presentations. I don't think we've got any more webinars lined up at the moment, but in your survey, if you want to indicate if there's anything else you want to hear about that we do, we'd be happy to try and put something together. So on that, I think we'll wind up there and just say thank you very much again to the presenters, and thank you all for joining.